There are some very significant differences between a virus and a bacteria. A virus cannot replicate outside of a cell, and a virus has only DNA or only RNA. It does not have both. And it has to use the cell machinery for its replication. The virus often brings in some enzymes that will help it replicate. That's enzymes of its own. And these have become the major targets of trying to find antiviral agents that will specifically work on the virus without having toxic effects in the cell. A bacteria can replicate on its own. It can grow in a petri dish. It doesn't have to have cells to grow in. A bacteria has both DNA and RNA, and antibacterials are directed at any of the machinery within the bacteria that is needed for the bacteria to grow. It can affect the cell wall, it can affect the cell membrane, and because bacteria have their own unique proteins involved in protein synthesis and nucleic acid synthesis, it has been easier in the past to target antibacterial agents or antibiotics than it has been for antiviral agents. I remember one of my first lectures on antivirals that was in 1967 in a conference at Banff, and it was supposed to be a 1-hour talk, and the gentleman got up and said, viruses grow within cells and they use the cell's machinery to replicate. There really is not going to be any good antivirals that will ever be specific, and I think it will be very difficult to ever get an antiviral. And that was sort of the concept until acyclovir was discovered by Gertrude Elion at Burroughs Wellcome. She later got the Nobel Prize for that, discovering a very specific antiviral agent. And since then, we have many good antivirals. And I would just use the example of AIDS. I have seen the development of AIDS from its very beginning. I had the first AIDS patient in Edmonton in 1983, and I've seen the whole spectrum of AIDS from the beginning to the present day pretty well. But when we saw the patients with AIDS in 1980s and right up until 1996, most of those patients we would see and diagnose, and we would see them deteriorate, and we knew that the average life expectancy was around 11 months before we had antivirals. Today we have over 20 drugs licensed to treat AIDS. Today, basic research, the discovery of the structure of the proteins in the virus, the unique proteins in the virus, such as the protease, have led to a whole group of antiviral agents. And today, we expect the life expectancy of AIDS patients to be right back nearly normal. And most of those patients are back working, useful lives, and we expect a minimum of 30 years and maybe a full life expectancy. And that is an example of how basic research, discovery research in a lab, has been translated into how you really benefit patients. And I would like to stress that without people toiling in the labs and doing the basic research and working out the structure of these proteins, we would not have the antivirals today that have changed that disease so much.